Hey guys, how are you today? Today we're going to start the fourth, I believe, the fifth part of the biology, inheritance and environment. This is the first video about mutations. So let's get right into it. First of all, what is a mutation? Such a great question. Any change in the genome is called a mutation. And you're talking about change. Does that mean that if I have something different from you, is that a mutation? Actually, not. It is not a mutation. But if the genome is supposed to be the same in all of your cells, and let's say one cell has a different nucleotide sequence than its neighbor cell, this is a mutation. So we're talking about the same organism. Or a mutation is something that is not the normal sequence. For instance, we will see right into it what mutations are through an example. But first, let's understand mutations. Mutations usually are due to mistakes during DNA replication that are not corrected. Oh, I forgot to mention something. Before you start watching this type of videos, you should have covered the IMAT material called reproduction and inheritance. As I have said in the past, I won't be making videos about this part of the biology right now, but after September 14th, you should expect to see them in my channel, the videos on reproduction and inheritance. But right now, I won't. I have passed this part, and you should have studied that on your own. Now let me continue. A mutation is any sequence of nucleotides in a DNA molecule that does not exactly match the original DNA molecule from which it was copied. But what I want you to know is that it can occur in the somatic cells or during gametogenesis in the gametes. Now, mutations that occur in the somatic cells are not being passed to the next generations, but mutations that happen in the gametes affect the offspring and not the parent. Okay, but mutations happen in somatic cells are way more often because somatic cells are this high of a number while gametes are much less. So if we want to see a ratio, it would be much more common to see mutations happen in somatic cells than in the gametes. Now, I want to talk about a specific part of a mutation in order to understand, and I want to give you a specific example and a specific disease. But before I do this, I need you to remind the structure of hemoglobin and red blood cells. What are red blood cells? They are sacs of hemoglobin. And what is hemoglobin? This is a red blood cell, this hemoglobin. It's a globular protein. It's made up of 4 polypeptide chains. 2 of them are similar, that means we've got 1 and 1 similar and 1 and 1 similar. Okay, and each polypeptide chain has a hem group with iron when the oxygen is being connected. So hemoglobin, the structure of hemoglobin is to transport oxygen. Now, there are many different hemoglobin molecules according to what chains make up the hemoglobin. So during the embryonic and the adult age, we've got different type of hemoglobins. In the embryo, the main hemoglobin is HbF, which has a structure of alpha 2 gamma 2. That means it has 2 alpha chains or alpha globins and 2 gamma globins. In the adult, we've got HbA, alpha 2 beta 2. That means we've got 2 alpha globins and 2 beta globins, which make up the hemoglobin. HbA2 in very small quantities, and we can find HbF in a percentage smaller than 1%. And these chains, AB, alpha, beta, gamma, and delta, are called globin. And altogether, this is the hem group, therefore altogether they make up the hemoglobin. And it's been written like that correctly, not like just in it, hem, because it has an A. Now let's look into sickle cell disease. Actually, sickle cell disease was the first disease that was found to be the result of a specific mutation. The researchers called Linus Pauling and his partners in 1949, he discovered that the hemoglobin of the adults, HbA, which is made up of 4 globins, 2A and 2B, 2 beta, 2 alpha and 2 beta chains, was different in normal people than in people with sickle cell disease or anemia. Today, we know that this difference is traced to in the 6th amino acid of the beta globin, where glutamic acid in the normal beta globin is replaced by valine. The beta globin, where the 6th amino acid is the valine and not the glutamic acid, is called an S globin. And hemoglobin with 2 alpha chains and 2 S chains, 2 alpha globin and 2 S globin, is called HbS. So people with sickle cell disease have HbS as their main hemoglobin molecule. Now, this difference in the sequence of amino acids is the result of a gene mutation in the triplet that codes glutamic acid. You see, in the antisense strand of the DNA, that is the sense that is not being transcripted, if one base changes and the normal codon GAG that codes glutamic acid, the A changes, is replaced by GTG, and GTG codes valine. This change, what is the result of this change? This changes the structure of the hemoglobin, which result in the change of the shape of the red blood cell. This is a normal red blood cell, and this is a sickle cell. So you see that the shape of the red blood cell changes. In low oxygen condition, it takes that shape. Now, those sickle cells, they obstruct the normal flow of blood in the capillaries, because you see, they don't have the correct shape in order for the capillaries. No, they don't have the correct shape in order to flow correctly into the capillaries. So they obstruct, they come here and they obstruct the capillaries, which cause problems in the blood circulation in many organs, like the lungs and the spleen. So this is the sickle cell anemia, a disease which is the result of a single mutation. Now, what types of mutation exist? Let's get going right into it. First of all, we've got gene and chromosome mutations. You see, if we're talking about 1 base, 2 bases, 3 bases, 4, 5, a small number of bases that change, then we're talking about a gene mutation because it happens inside a gene. But now if we're talking about a large part of the chromosome where we could have, I don't know, a whole part of the chromosome deleted, that is a chromosome mutation because it takes place in much larger parts of the chromosome. So according to the extent of the mutation, we've got gene mutation and chromosome mutations. Let's look into it, gene mutation. We've got 4 kinds. Let me show you the 4th kind. First of all, this is the normal DNA sequence of the sense strand. Let's see substitution. We see that the C here, 1 base, is being substituted by T. If that happens, you can see that the C is being substituted by the T. And therefore, the mRNA codon GAA has now become AAA. And AAA does not code for the same amino acid than GAA, and it codes for AAA least. That results in a different amino acid sequence. And this is substitution is what we had in sickle cell anemia, if you remember. So sickle cell disease, we've got the replacement of G of A by T, and we had as a result valine. And we saw how important the change of only 1 out of the many millions of DNA bases has what effect. And then we have insertion. In this type, we have to add here, in this place, another base. This is bad. You know why this is bad? Because it happens here, the frame shift mutation, but I won't go there. Let's imagine now that I add 3 base pairs, not just 1. And therefore here I have 3 base pairs, therefore I would have another glue. Okay, and we've got again a change in the amino acid. And last but not least, we've got deletion, and let's imagine I delete these 3. They are not being replicated. So the final, I delete these 3, the final mRNA will be Fe, Tir, and then glue, not this glue. This will go away. Okay, but sometimes when that does not happen, when insertion and deletion does not happen in the number of 3, or 6, or 9, or 12, we have something else called frame shift mutation. This displaces all the nucleotides to the right, to the left, it doesn't matter. And it changes the reading, how we read the DNA. And we've got a completely different sequence in the amino acids now. The amino acid sequence has no similarities than the first one. And here you can see we've got this sequence, and I add, it's not that I change the frame shift. It starts reading from here, not that. You know that we have, you know the characteristics of the genetic code, and therefore you should know how to read that. So it starts from here. Now we've got a completely different, look at that, a completely different amino acid sequence. Now, what is the result? I didn't write a PDF file here. I just screenshotted a part of the book that says it excellently. So, it says that a silence mutation occurs when the new codon still codes for the same amino acid. And that's called a silent mutation, because a mutation takes place but we see no result, no. And how could that happen? If I substitute this T with C, CTC creates GAG, and as you see here, GAG codes glue. This is due to a characteristic of the genetic code. Okay, that many codons code for the same amino acid from 2 to 6, with 2 exceptions. Tryptophan, I don't know how it's in English, and tryptophan, and give me a second, oh my god, tryptophan and methionine. Yes, these 2, methionine and tryptophan, are being coded by only 1 codon. All the others are being coded from 2 to 6. Okay, and if we've got that and it change is a synonym code, as it is called, codon, a synonym codon, then we see a silent mutation. Then we've got a missense. We have a new codons for a new amino acid, like here. And last, we've got a nonsense mutation, when the new codon happens to be a stop codon. Okay, I want to give you an example of that. Now we've got chromosomal aberrations. What type we may have? We may have deletion. When we've got, let's say this is the chromosome, the normal chromosome, and after deletion I get this. This was here, and now this is missing. We've got a deletion. I may have an inversion. What is that? This is actually a quite complex one. So let me show you something. If I have A, B, C, and D, how inversion happens? The chromosome is being cut in 2 places. Let's say here in C and in B, and this is reconnected with reverse orientation. That means I would have A, So I take that, I turn it around, and C comes here, B comes here, and I got D normal. This changes the sequence of the genes in the chromosome. Then we've got translocation. Translocation, we've got actually two types. This is a pair of homologous chromosomes. Where is my... And this is another pair of homologous chromosomes. If a part from the chromosome here is being cutted, let's say I cut this one, so it goes from here and it connects here, this is a translocation. But another thing is called a mutual translocation, I believe so. What's happening? I cut this part here and I cut this part here. And this is being exchanged with this. These are DNA sequence of different chromosomes, non-homologous. And this is mutual translocation. This does not have any result in the organism, but if it happens in a gamete, then the offspring has a high chance of an irregular chromosome number and structure. And then we've got this that I'm not going to talk here. I'm going to see here. There is a thing in chromosomal aberrations which is called Nondisjunction. It is an error that sometimes occurs during meiosis, in which homologous chromosomes fail to separate as they should. Let's see an example. I can have two types of nondisjunction. If we have an error during meiosis 1, and I don't have the separation of the homologous pairs, and therefore I got 2, and if they're connected with another one, what happens? I've got 3 chromosomes. Therefore, this pair would have 3, and from 46 I would go to 47, and this is called trisomy. For instance, people with Down syndrome, they have an extra chromosome 21, or they can be monosomic. This is only found in Turner syndrome, where we've got only one sex chromosome, the X, no other, and we got 45 chromosomes overall. And then Klinefelter, we've got a trisomy in the sex chromosomes, where there is an extra X chromosome. People with Klinefelter have male genitals, but the testes are abnormally small and the men are sterile. And the Down syndrome, people with Down syndrome have characteristic facial features, mental retardation, prone to developing Alzheimer's and leukemia. And why a mutation created? First of all, they can be created from replication errors or from mutagens. These are environmental factors that cause mutations. What substances are mutagens? What environmental factors are mutagens? Some chemical substances, like this here, or some types of radiation, like X-ray, gamma ray, ultraviolet, and cosmic radiation. And the mutagens that activate uncontrolled cell growth is cancer, are called carcinogens. It are the mutagens that cause cancer, activate uncontrolled cell growth. That is all I believe you should know about mutations. Thank you for watching my videos, guys. Subscribe to my channel here, and that's all. Bye bye.